Hello and welcome to the final longevity review of 2024. This month, we will take a look at gene therapy to increase telomere length, freeing the immune system to remove senescent cells, using probiotics to help with sarcopenia, and in the Canadian content study, employing extracellular vesicles to deliver drugs to the brain to combat neuroinflammation. This review is presented to you by the Canadian Longevity Association, Canada's premier longevity nonprofit. And my name is Chris Linnell, the founder and president of the association. If you'd like to find out more about what we do, then please check out the links below this video, where you can also find links to the newsletter and studies reviewed here. And lastly, don't forget to like this video and subscribe to our channel. So let's take a look at the first study. In this study, researchers used gene therapy to lengthen mouse telomeres. Now, telomeres, if you remember, are the protective caps at the end of our chromosomes, which shorten as we age and eventually leads to senescence and other dysfunctions. 20 years ago, this process was thought to be the main driver of aging. And the idea was that if you could lengthen them, then lifespan could be increased. Telomeres can be lengthened by increasing the expression of telomerase reverse transcriptase, or TERT, and it is encoded by a gene of the same name. All of the methods used to increase the length of telomeres have involved increasing the expression of TERT. Now, they have fallen out of favor in recent years. However, there have been several studies which have shown that if you could lengthen them, then lifespan can be increased by up to 40%. Those studies use viral vectors, while this one takes a slightly different approach. Now, I should straight away say that the method used in this study is not really applicable to humans, as you will shortly see. However, as a proof of principle and the results shown, it's certainly worth checking out. So let's take a look. I won't go into too much detail as to how they did this, but basically they took mTERT, which stands for mouse TERT, and they implanted it into embryonic stem cells from black 6 mice, and those cells were enriched, and then they were injected into a blastocyst stage of the embryo of other mice, created chimeric embryos, which were then implanted into surrogate mothers. And then various sort of matings took place to create mice which always overexpressed TERT. And because they were placed in the germline of the mice, they were then passed down, or then TERT was passed down to succeeding generations. And in fact, they used 5 generations of mice in this experiment. So you can sort of see that this is not really applicable to us, or at least us living humans. And I'm not sure we really want to do this for our unborn or yet to come humans. But again, this is an interesting proof of principle, as you will shortly see. Once they had done that, they obviously wanted to see that it was effective. And firstly, they looked at the mRNA levels of TERT in various organs, so lung all the way to skin here. And the orange are the wild type, or the control mice, and the blue bars are the treated mice. And you can see here that the expression of TERT was higher in all of the organs, quite considerably, in fact. If you look here above, you can see the p-values. They're very low, which means that they are highly significant changes, which you can also see just from the eyeball test. And that was the mRNA. Now, the actual telomerase activity also was substantially higher, hugely higher compared to the wild type mice. You can see the blue bars are way higher than the orange bars. And in fact, when they looked at the length of the telomeres, they were also quite a bit higher than the control mice. And for the most part, they also reached statistical significance. Only in the spleen did it not quite reach significance. So this showed that the therapy worked, at least in terms of lengthening telomeres. Of course, we're not interested in lengthening telomeres for its own sake. We want to see what effect it had. And that effect was quite substantial, as you'll shortly see. So here are the survival curves of all 5 generations of mice. Now, in A, here are the combined results of all 5 generations, and the black line are the wild type mice, the control mice. The gray line are mice which were given gene therapy without the TERT gene, and the orange line are the treated mice which were given the TERT gene to overexpress telomerase. And you can see here, compared to the wild-type mice, the median lifespan increased by 16.57%, whereas the maximum lifespan increased by 27.48%, a quite considerable increase. Now, I should just mention the researchers say that the control mice lived slightly shorter than the average black 6 mouse lifespan. However, that shortening was probably because of the specific housing conditions of those mice, but that should also have affected the treated mice. Now, from B to F are the 5 generations of mice, starting with the first one with B, where they had a 21% increase in median lifespan and a 15% increase in maximum lifespan. Second generation had a slight reduction, or considerable slight reduction in median lifespan of 19% and a considerable reduction in maximum lifespan, only 2.75% more. However, the third generation had a whopping 49.54% increase in median lifespan, which is actually the largest increase in median mouse lifespan that I have seen in a study about treated mice. And they also had a 27.48% increase in maximum lifespan. Those numbers sort of reduced for the fourth and fifth generations. As you can see, going from 20% median lifespan with the fourth one and 11% for the fifth one, and the maximum lifespan going from 11% for the fourth generation and 21% for the fifth generation. So all in all, quite impressive. Now, looking at it from a male and female point of view, you can see here that they were pretty much identical. Maybe the females lived slightly longer. Now, the one thing to note is in this strain of mice, the original mice, the black 6, males tend to live longer than females. So this treatment at least got rid of that difference. H here is once again just showing the expression levels of TERT in the liver, which led the researchers to look at the antioxidant activity, or the endogenous, or the antioxidants which our body produces, specifically glutathione and superoxide dismutase. And for both of these antioxidant enzymes, the levels were quite considerably higher in the treated mice than in the controls. Once again, the treated mice are in blue here. And looking at p53 expression in the liver, and p53 is a gene which suppresses cancer, but also is a senescent marker. And here, the levels of p53 expression were once again significantly lower in the treated mice than in the controls. Again, another good thing. The researchers didn't just look at lifespan, they also wanted to see what the regenerative capability of the TERT overexpressing mice was. And they did so by looking at wound healing in the skin and also in the colon. Now, firstly, in A here, it shows levels of TERT expression in the skin. And once again, there was a significant increase in the treated mice. Now, the poor mice were given puncture wounds, though they were under general anesthesia for this, so they shouldn't have felt any pain. And you can see the pictures here of those puncture wounds over the course of 7 days of healing. The top are the control mice, the bottom are the TERT mice. And you can see here just from the eyeball test that their wounds healed quite a bit faster. And there was also faster hair growth in the treated mice. And we can see here on the right is the wound healing rate in mice percentage. And once again, the blue bars are the treated mice, and their wound healing rate was higher than the control mice. And for the most part, that reached significance as well. Next on C here were levels of hydroxyproline, which is one of the main constituents of collagen, which is the main protein in skin. And once again, the levels were higher in the treated mice than in the control mice, though at the end, after 10 days, it did not reach significance. You can see by the p-value. On D here, it shows how the skin cells migrated. So they would have to migrate to sort of cover the wound. And once again, the wound healing rate of cells, or the migration rate, was higher in the treated mice than in the controls. Looking at the molecules involved in this process, in E here, The top two are inflammatory molecules, or at least their mRNA levels. And when we are injured or wounded or even sick, inflammation rises starkly at the beginning before sort of subsiding. And it appears here that compared to the wild type or control mice, that the inflammatory molecules were much higher at the beginning before falling to levels below that of the wild type mice. And their levels are this dashed pink line you can see here. The bottom three are mRNAs for growth factors, the first one for fibroblasts and the second two for collagen. And here the levels were always higher than that of the control mice. Again, the blue bars are considerably higher than this dashed pink line. And on the right here, we have the expression of three protein growth factors, FGF7, which is the same as on the left here, VGF and WNT1. And again, once again, for all of these, their levels were higher than that of the control mice at all stages of the wound healing. So this basically shows that the mice, well, you can see in the pictures, the mice healed faster, and this was obviously shown by the images up here, but that was backed up by these increased levels of growth factors. So overall, the mice had much better wound healing capacity, as can be seen by the photos, which was then backed up by these growth factor expression levels. So this is obviously quite positive. Lastly, the researchers looked at the regeneration of the colon after colitis had been induced via dextrin sodium sulfate. Now, once again, starting by looking at the levels of TERT in the colon in A here, and we can see once again that the levels were a lot higher in the treated mice than in the controls. Next, in B here, we can see the actual colons of the mice, and during colitis, the colon supposedly shrinks, and how much it shrinks indicates, I guess, how much inflammation or how much damage has been caused. And on the left are the mice which hadn't been given colitis, and on the right are the mice which had been given the DSS-induced colitis. And basically, it seems that the colon of the treated mice did not shrink as much as that of the untreated wild type mice. And that can also be seen visually in these pictures, but also can be seen here on this sort of bar graph. And the darker red and darker green are the untreated ones, and the lighter pink and lighter blue are the colitis mice. And the wild type mice had a much greater shrinkage of their colon, comparing these two, compared to the treated mice, who, although the colon did shrink, it did not shrink as much, which is seen as being positive. Next, they looked at the score of loose stools. I guess colitis causes loose stools, and the levels were much lower in the treated mice than the controls. However, the score of bloody stools wasn't significantly different, although it looks a little bit lower here. Yeah, it's certainly, looking at the p-values, did not reach significance at all. However, looking at cross-sections of the colon, and again, on the left are the mice which did not get colitis, control mice at the top, treated mice at the bottom. On the right are the mice which were given colitis. And basically, according to the authors, there was less cell death, there was less edema, so like bleeding, there was less inflammatory cell migration, and less vessel dilation, all of which are good things. I guess just looking at from the eyeballs here, there's more white area here than here, but again, I'm not an expert in colon dissection. Now looking at E, and I'm going to make it a bit bigger so you can see, here were three proteins, again used, I guess, in the regeneration of the colon, and levels were different between the control mice and the treated mice for all three of these proteins. And the authors didn't really go into detail, but they said these differences were positive. So there you have it. Not only did TERT overexpression improve healing, both in the colon and the skin, but it considerably lengthened the mouse lifespan. Now, as I mentioned at the beginning, the method used in this study isn't really applicable to humans, but however, it is a proof of concept, and again, demonstrating potential powers of lengthening our telomeres. And the authors mention this in this study of a previous study in July of 2024, which I hadn't seen because it was behind a paywall. However, researchers have discovered compounds which can lengthen telomeres successfully in mice, and there are supplements you can buy which supposedly lengthen telomeres as well. I'm not suggesting you try those. However, it does seem possible that we will soon have actual compounds or drugs which could lengthen our telomeres without having to use gene therapy. Anyway, this is a very encouraging study nonetheless. Moving on to the second study. This paper looks at a new way of removing senescent cells. Now, a reminder that senescent cells are dysfunctional or zombie-like cells which accumulate with age and cause various problems in our bodies. And removing them is generally a good thing. Up until now, most methods used to remove senescent cells have involved either finding molecules which can trigger apoptosis, which is cell suicide, or using CAR T cells to directly kill them. However, our immune system should theoretically clear them out, yet it does not do so and lets them accumulate. The researchers here found a way to remove the shackles from the immune system and let it get on with its job of clearing senescent cells. So let's take a look at how they did so. Okay, so I'm not going to go into too much detail for the first few figures, but you'll get the general idea of what they found out. So firstly, they wanted to take a look at whether or not senescent cells actually stop the immune system from properly activating. And the method that the immune system would use to kill off cells would be by using natural killer cells, or NK cells as they term here. And those natural killer cells would find the senescent cell or cancerous cell or some other pathogen, and it would fire sort of pellets or shotgun pellets at the offending cell, thereby killing it. And that process is called degranulation. And what they firstly did to see how this worked was they injected mice with proliferating normal cells or senescent cells. And what they found out was that, so the green are the sort of normal cells, blue are the senescent cells. They found out that the NK, or the natural killer cells, did sort of home in on the senescent cells more so than the normal cells, as sort of can be seen here by the higher blue dots to the green. However, there was less degranulation, so they weren't sort of activating or attacking those cells, as can be seen in E here, where the blue dots are lower than the green ones. And in a second set of experiments, they found pretty much the same thing, whereby there was less degranulation in G and H here. Again, you can see the blue is below the green, and also less killing of those cells. Again, the blue is much lower than the green here. Next, they looked at what was causing this. And basically, they found out that it wasn't caused by anything that the cells were releasing themselves. So senescent cells release a bunch of molecules known as the senescent-associated secretory phenotype, or SASP. And they found out the fact that the natural killer cells weren't being sort of activated was not caused by that. Instead, they found out that it was being caused by a receptor on the surface of cells called ganglioside-GD3. And this GD3 only became prevalent once cells were senescent. And the next few figures sort of delve more deeply on this, but though we will sort of skim over it quite quickly. In figure 3 here, they found that when senescence was caused by a cancer gene or oncogene-induced senescence, GD3 expression did not arise. So sort of an interesting insight or something, though I'm not sure how relevant it is to us. Next, they carried out various other experiments to show that it was indeed GD3 expression which caused the natural killer cells not to go into a proper attack mode. And then they looked at the expression of GD3 in various senescent diseases. The first of which was bleomycin-induced lung fibrosis. Now, bleomycin is a potent and toxic antibiotic that gets used in cancer treatment and can also cause fibrosis in the body. And so they use that to simulate a lung fibrotic disease. And you can see here, just in B at first, that levels of collagen. So fibrosis is quite often caused by, or mainly caused by, the accumulation of excessive collagen, which causes the fibrosis. And there was a lot more collagen deposition in the bleomycin treated mice in blue here compared to the controls. And I go also in the staining here on the left, there was much greater staining, I guess, collagen staining in the bleomycin induced mice. And when they looked at that more closely, and see here at the top, you can see SA beta gal is a marker for senescence. So you can see that there were a lot of senescent cells. And looking inside of those cells, there was a lot of GD3 expression. You can see, I guess, by the red here, sort of zoomed in on the right from this white square here. They then took those same natural killer cells and re-challenged them against something called YAC, and there was less degranulation as a result. However, cells taken from the spleen showed no such drop. And so they think this was caused by the GD3 in the lungs. Now, over the period of time, lung fibrosis decreases. And you can see by the staining here on the left that the red or purpley red gets less. And you can see here in G on the left that the amount of collagen rises sharply and then decreases again. However, at the same time, levels of GD3 didn't change that much. You can see that it increased here quite a bit and sort of stayed elevated compared to the black control here, or the day 0. And on these images, I guess the red dots are again the GD3, and they persist more than the collagen does. They also looked at liver fibrosis caused by a Western diet from H2K here. And once again, the staining shows increases in collagen levels. So see an I here, collagen area increased. P21 cells are a marker of senescence, and once again that increased quite a bit. And J shows the expression of genes which cause collagen at the top, or produce collagen at the top here, showing a big increase. And this ST8SIA1 is, I think, the gene encoding GD3, again showing a marked increase. Lastly, on this figure, they also looked at kidney fibrosis caused by something called ADR, and at L here on the bottom left, once again, this is showing the senescent cells in the fibrotic kidneys, and the GD3 levels were quite high as well. And then on the right here is showing the percentage of the kidney area covered by GD3 signals, and compared on the left are sort of mice which were just given salt, and on the right mice which were given this ADR treatment, I guess, which causes fibrosis. And this is the total area at the top of the kidney, and the bottom is the sort of filtering part of the kidney. And again, levels of GD3 were very much elevated there. Skipping over the next figure, which was simply further analysis and proof that GD3 is associated with senescence as a surface marker on the cell, we get to the interesting figure, because the researchers didn't just find out that GD3 was stopping natural killer cells from attacking senescent cells. They found that by using an antibody to block GD3, they could unleash the natural killer cells to attack the senescent cells. So starting at A with the study design. So first the mice were given bleomycin to cause the lung fibrosis, and then 7 succeeding days they were given this anti-GD3 treatment and then were euthanized and examined. Now before they were euthanized, the survival curve in B here can be seen. So blue line are the mice which were not treated, and the green line are the mice which were given the anti-GD3 therapy. And you can see that survival was markedly more. I guess they died at less than half the rate. You can see the survival curve kind of sort of flattens out. And if you remember from the previous figure, fibrosis sort of abates, gets better after a while. So I guess the mice stopped dying, but the survival rate is significantly better. Now looking at C here, which is the amount of collagen that is deposited, and you can see that the treated mice has significantly less collagen put down on their lungs. And in D here is showing the levels of senescent cells according to SA beta gal. The treated mice had a lot less senescent cells than the untreated mice, and they had higher levels of the natural killer cells than the untreated mice as well. Looking at F here, you can see the level of, I guess, collagen deposition, the level of fibrosis. And the blue lines, once again, are the control mice. The green lines are the treated mice. And for the most part, apart from looks like one one unlucky treated mouse, they fared a lot better than the control mice. The lines are either going down or not rising as steeply as the untreated mice. And I guess can also be seen in these images as cross sections of the collagen deposition. Here on the left are the untreated mice. On the right are the treated mice, and they have less collagen or less red, you can see in these two images. In G here, we can see the levels of GD3. And after 14 and 21 days, the green treated mice had much lower levels of GD3 than the untreated mice. And in HP16, once again, is a marker of senescent cells, and it was lower in the treated mice than in the non-treated mice. In the last experiment, the researchers gave this anti-GD3 treatment to old mice without inducing any disease. So the mice were 18 months old, which is roughly equivalent of a human in their 50s. And over the course of 3 months, they were given 6 injections of this anti-GD3. And they took a look at age-related lung, liver fibrosis, as well as age-related osteoarthritis. And what they found in the lung was that once again, the collagen deposition was a lot less in the anti-GD3 mice, and the levels of GD3 were, unsurprisingly, a lot lower, apart from this one mouse up here. And the same could be seen when they took a cross-section of the lungs. There's less red staining for collagen in the anti-GD treated mice. And the same thing could be seen in the liver, where once again, in the cross sections there was less collagen, I guess. And also on the right here we can see that levels of collagen were a lot lower in the treated mice than the controls. Lastly, they looked at osteoarthritis, and the bone volume was higher in the treated mice. You can see also on these images, I guess there's not as many gaps in the treated mice on the right here compared to on the left, but you can see on these graphs that the green triangles are higher than the blue dots, and that's to a significant level. And also the thickness of the bone is higher in the treated mice, and the bone surface area to volume is also lower, which is a better thing for the mice. So you can see that anti-GD3 therapy showed improvement in various age-related conditions. So that's the end of this paper, and it's still very early days for this type of treatment. I don't know if anti-GD3 therapy as used in these experiments can be used in humans or not, but it's certainly a good starting point for therapies which could be used against GD3 and activate our immune system. And it's nice to see that we have another bullet in our senolytic armatorium. Now to the third study being reviewed. In this paper, researchers used beneficial bacteria, or probiotics, to help treat sarcopenia. Sarcopenia is age-related muscle wasting and can lead to frailty and ultimately mortality. Therefore, it is an imperative to find a treatment for it. Although diet and exercise can help, unfortunately, they're not beneficial for most people. In this study, a strain of Bifidobacterium animalis, Probio M8, was used. This strain has already been tested in people for Parkinson's and osteoporosis. In Parkinson's, it improved sleep quality, anxiety, and gastrointestinal symptoms, while in osteoporosis, it improved bone metabolism. Let's see what effect it had on sarcopenia. First, they tested it on mice. They gave 19-month-old mice, which is the equivalent of someone in their late 50s, Probio M8, I'll call it PM8 from now on, for 28 days before sacrificing the animals. The control mice were just given a salt solution instead. On the right in B here, you can see that the weight of both the control and the PM8 mice decreased over the course of the study, but there was no real difference between the two. The Probio M8 mice started off with a lower weight, and that was the same towards the end. There was no significant difference. However, when looking at senescence scores, and C here on the left, there was a significant difference between the controls and PM8 mice. So the PM8 mice are these blue circles, the controls are in the dark red. and there was significantly less senescence according to their metric. Now, pulling here in D means sort of grip strength, how much they could maybe hang from the wire, and the PM8 mice had greater strength or greater pulling ability, which was also significant, as you can see by this star here. Now, next on the right here, distance to exhaustion, so how far they could run, and the PM8 mice ran a lot further, as you can see here. Next, looking at the composition of the microbiome in E on the right here. In a nutshell, the control mice had an increase in abundance of pathological bacteria, whereas the PM8 mice had an increase in beneficial species. Looking at the fecal metabolome on the left here, there was an increase in beneficial metabolites such as L-selenomethane and lipoxin B4. So lipoxin B4 is an anti-inflammatory and L-seleno is cancer preventative. And in the serum metabolome, L-serine, carnosine, and coenzyme Q10 were increased in the PM8 mice, which was also beneficial. This can more precisely be seen in G here, where levels of L-selenomethionine increased. Lipoxin B4 increased as well. LTB4 went down. Now, unfortunately, they don't mention in the text what LTB4 is or does, but I guess lower levels hopefully is beneficial. And below here, L-serine, which leads to creatine, carnosine, and coenzyme Q10 were all significantly higher in the PM8 mice. However, we are more interested in humans than in mice, and luckily that is whom they tested PM8 on next. So this was a small study with 43 sarcopenic patients, and they were treated with PM8 for 60 days and luckily not sacrificed at the end of that. And what they found out was the following. Starting on the left here, ASM height shows skeletal muscle mass, and there was no real difference between day 0 and day 60. So just to decipher this, they divided it by females and males. Females on the left, males on the right. The red dots represent day 0. The blue dots represent day 60. And unfortunately, there was no real change. The sort of same goes with grip strength. There was maybe a slight increase, but it certainly did not reach any statistical significance. However, with the FTCS, which stands for the 5 times chair stand test, so how long it takes for somebody to stand up 5 times from a chair, and here there was a significant difference between day 0 and day 60, with a 16% reduction in the time taken to accomplish that fact. Improvements in this metric can make a measurable difference to someone's quality of life. As for the rest of the measures, calf circumference did not change, and nor did BMI, triglycerides, HDL, cholesterol, LDL cholesterol. However, total cholesterol did decrease significantly. As for blood glucose, albumin, globulin, prealbumin, and C-reactive protein, there was no significant change between day 0 and day 60, though it does look like globulin decreased, though the authors didn't say anything about that. Next, the authors looked at the microbiome of the participants, and what they found was that there was a limited impact in terms of the richness of the microbiome by looking at the Shannon and the Simpson index. There didn't seem to be much change there, but there appeared to be a shift in the composition of the microbiome, as shown by this supposedly. And looking at individual species here, just like with the mice, pathological bacteria decreased, whereas beneficial bacteria increased. And looking down at E here, which looks at metabolic pathways with dots to the right showing a greater increase or change, we can see that vitamin C biosynthesis is one of the pathways which increased markedly, which is a positive considering that vitamin C is beneficial. Next, they looked at the fecal and serum metabolome, and just like in the mice, there was an increase in beneficial metabolites, so things such as indole-3 lactic acid in the fecal metabolome is an anti-inflammatory, and something like acetoacetic acid promotes the proliferation of muscle cells, supposedly. In the serum metabolome, it was also noted that creatine increased, and creatine is very beneficial for muscle function. And looking more specifically at creatine, we can see that the levels of creatine increase between day 0 and day 60, according to the scattered graph here on the right. And although it doesn't look like much, it did reach statistical significance. Now, HSL on the left here is a molecule produced by some bacteria which blocks the uptake of creatine, and its levels declined over the course of those 60 days, also reaching significance. Lastly, they looked at this HSL creatine connection via an in vitro model, and basically, in a nutshell, found that PM8 reduces HSL, which allows for a greater uptake of creatine into the body, and that proves to be beneficial for those sarcopenic patients. Now, ending on their little graphical abstract here at the bottom. So the authors conclude that ProBio M8 regulated critical microbiota. It inhibited the enrichment of HSL in the gut, thereby alleviating the accumulation of serum creatine and improving somatic function in the host. In layman's terms, basically, ProBio M8 changed the gut microbiome and stopped the bacteria producing, or reduced the bacteria producing HSL, which meant that more creatine was taken up into the body, which turned out to be beneficial for the muscles and thereby reduced their 5 times chair stand test. Now, of course, the question is, could they have just taken creatine on its own? Would that have made the same bit of difference? Who knows? They didn't test this out. Now, this study isn't earth-shattering. However, considering the prevalence of sarcopenia, anything that can help alleviate it is beneficial. And considering how simple it would be for patients simply to take a probiotic capsule, this could help alleviate the scourge of sarcopenia, or at least make a small difference towards that goal. And finally, the Canadian content study. Coming from the lab of Charles Ramasamy and lead author Rumonegi Oliveira Silva of the Armand Frappier Santé Biotechnology Research Center, this study looked at using extracellular vesicles to deliver a drug payload to the brain to combat neuroinflammation. Now, extracellular vesicles are small spherical packets of information released by cells to communicate with each other, and they show great potential in treating diseases as well as aiding in longevity. Let's start by taking a look at the graphical abstract. Starting at the top left, they had 2 sources of extracellular vesicles, or EVs. One was human plasma, and the other one was adipose mesenchymal stem cells, basically stem cells that are found in fat tissue, and they were sort of grown in culture. Anyway, they got the EVs from there, and you can sort of see what an EV looks like. I guess sort of cross-section, sort of spherical, and has all sorts of receptors on the outside, and various chemical sort of or contents, including DNA and various other things. Anyway, they loaded those with donepezil, which is a drug used to treat mild to moderate Alzheimer's disease, and it is also an anti-inflammatory. Anyway, they loaded it into the EVs, as you can sort of see here. Then they were injected into the animals. In this case, it was zebrafish larvae, where they entered circulation and were able to cross the blood-brain barrier, which is quite difficult for a lot of drugs. It stops around 95% of molecules from entering the brain. However, EVs can do so, so that's why they are a great payload carrier. And once into the brain, they were taken up by the cells, with, it says here, quickly internalized, and they showed no toxicity. And they also prevented the migration of pro-inflammatory microglia, which are immune cells which can also cause inflammation. And when they sort of stopped those immune cells from migrating or causing inflammation, they reduced all of these inflammatory molecules, such as IL-6 or TNF-A alpha, and also reduced reactive oxygen species, better known as sort of oxidants. If you think about antioxidants, they are sort of designed to combat ROS. And this resulted overall in lowered inflammation and oxidative stress, as you will shortly see. So let's take a look at how this study was done. So firstly, they characterized the size of these EVs. Now, they're labeled in this paper as PEVs to indicate the plasma EVs, and ADMSC to indicate the fat-derived EVs, and I'll just call them fat EVs from now on. So you can sort of see their size according to nanometers. So the fat EVs were similar in size to the plasma EVs. The sort of scale goes in both directions, and here you can see microscopic photos of them. They kind of look a bit like red blood cells to to my eyes at least. Anyway, moving on, they also then checked out their toxicity, and in A here it basically showed that they didn't have any toxicity, sort of 100% in terms of killing cells. And also they had a rather quick uptake into cells. This was in vitro, and they used microglia cells to see how quickly they absorbed or internalized these EVs. And here you can see from 1 hour, 24 hours, blue and green represent the EVs. Blue is a plasma, green represents the fat EVs. And after 24 hours, it reached over like 99% absorption. The purpley bar just represents some fluorescent beads of similar size to act as a control. And they were also taken up, but at a much slower rate. And here they're once again just looking at the size of the EVs once they are inside of those cells. Next, they tested out their anti-inflammatory potential in a test tube where cells were challenged with LPS, which is lipopolysaccharide, which is a molecule found on the surface of bacteria and which causes inflammation, basically. And what they found was that inflammation was reduced. So you can see from B to G here, these are all different inflammatory molecules. And in each of these charts, you have the black control bar, then the gray is just LPS given to the cells, and then the blue bar is the plasma EVs, green bars are the fat EVs. And you can see for each of the various molecules, you can see IL-6 here, TNF-A alpha here, some of the better known inflammatory molecules. And in each of these cases, the inflammation was reduced when compared to the LPS given on its own. Now, in all of these, apart from IL-6, the fat EVs perform better than the plasma EVs. You can see the green bars are slightly or significantly lower than the blue bars. However, they all perform better than, as I say, LPS given on its own. Next, they actually gave them 2 live animals. In this case, as I mentioned before, zebrafish larvae. And these zebrafish larvae were injected with the EVs 48 hours post-fertilization. And they were also tagged with fluorescent markers, so you could see sort of the distribution of these EVs. On the left are the plasma ones, on the right are the fat ones. And basically you can see that they pretty much went everywhere in the fish. And here's sort of a close-up of the brain. So it's quite bright, so they got pretty much everywhere. And the charts, the graphs on the right-hand side sort of show how much sort of got out of the blood vessels into the actual brain itself. And so those are quite big, a good distribution of them in the brain. So it shows that these EVs can disperse rapidly within the organism. Next, they wanted to see how well the EVs were taken up by the microglia. And from the side in B and C, and from the top in E and F, with the plasma EVs on the left-hand side again, and the fat EVs on the right-hand side, you can see with these sort of white arrows, here, it's slightly harder to make out. This indicates that they were taken up by the microglia, which is what they wanted to see. Then it came to adding the drug payload. As I said before, is donepezil, which is a widely used drug to try to treat mild to moderate Alzheimer's. It's not a cure in any way, shape, or form, but it reduces some of the symptoms and is better than nothing, I guess you can say. However, it also acts as a general anti-inflammatory. So just actually make this one a bit bigger and clearer, you can see in A here on the top left how they sort of added the donepezil into the EVs, and then they checked out the size of them once again, and various different sizes of them. And more importantly, they also checked out the toxicity when they were given to cells, and basically the toxicity was the same, if not better, compared to donepezil on its own when given to the cells. And on the right-hand side, we can see the uptake of these EVs as well as donepezil on its own into the cells. So the gray line is donepezil, so it was just released into the cell environment, and they took it up very quickly, and the EVs were taken up more slowly, and after 24 hours it reached around 80%. Also to mention that the encapsulation efficiency, how well the EVs took up the donepezil, was roughly 60%, which was also considered a good number. Next, They looked at the impact on phagocytosis, which is when microglia or any cell basically engulfs another cell or takes something inside. So one way that our immune system sort of kills off bacteria. And what they found was that there was sort of a lower level of phagocytosis with the EVs. Yeah, they just sort of say it was interesting that it was lower. Don't really mention if it was a good or a bad thing, but we'll leave it there. Now we move on to the end results. So firstly here, we have the results of the donepezil-loaded EVs, both in vitro, so in a test tube, and in a living organism. And you can sort of see the two graphics is how they did that on the top here. And B and C show sort of in vitro in a test tube against these microglia cells. And basically, it's hard to see in sort of B here, but in C, you can see the level of inflammation. Oh, also as well, the cells and the animals were also given LPS from this inflammatory causing agent. And the control is the black line here. If you look at the bottom, the gray bar is the cells which were just given LPS, so they had more sort of inflammation. Given donepezil on its own, lowered the inflammation. However, given the donepezil by the EVs, lowered the inflammation even more, with the fat EVs performing slightly better than the plasma EVs. Moving on to the live fish. So once again, they were also tagged with a fluorescent marker. And at the top here are the control fish, so you can see they only had a little bit of inflammation. Then next are the fish which are only given LPS, and you can see a lot of inflammation throughout its body. When they were given donepezil on its own, and you can see that the inflammation was reduced. However, when they were given donepezil inside the EVs, the inflammation was markedly reduced, especially for the plasma EVs. There's hardly any inflammation comparable to that of the controls. More so in the fat-derived EVs. However, it's still really reduced compared to the control or the donepezil on its own. And we can see that in these two bar charts at the bottom. Again, the control is black. The dark gray is LPS on its own. Light gray is donepezil on its own. Light blue is the plasma EVs. Greeny blue is the fat EVs. And you can see that the plasma EVs have lowered the inflammation by the greatest degree. And E is sort of the whole body. F is more concentrated in the brain area. Looking more closely at this anti-inflammatory effect. So here they wanted to see the effect of the inflammatory microglia sort of migration when they sort of moved to the sites of inflammation. And the zebrafish were injected with LPS again to cause inflammation, and then either given donepezil on its own or together with the two EVs. And sort of control here, you can see there's hardly any inflammation. When LPS was given on its own, there was quite a lot here, seen by the red. Donepezil didn't sort of really make much of a difference, at least visually here. However, for both of the EVs, and especially the fat EVs, there was a marked reduction, which we can also see in this bar chart here under C. So again, black is the control. LPS had a major increase in inflammation, unsurprisingly. Donepezil reduced it slightly, and still to a significant degree. However, the clear winners were the two EVs, especially the fat EVs, which reduced it by the greatest extent. Now on the right here, we have acetylcholinesterase activity. So acetylcholinesterase is a molecule which breaks down acetylcholine, which is a neurotransmitter. So anti-Alzheimer drugs like donepezil basically are an anti-acetylcholinesterase inhibitor, and they basically want to stop it breaking down this neurotransmitter. So we sort of want lower levels of acetylcholinesterase. And so in E here, you can see just control. Then donepezil doesn't sort of make much difference sort of on its own. However, the EVs, both the plasma EVs and especially the fat EVs, reduced the activity of acetylcholinesterase to a greater degree. Finally, they wanted to see what effect this had on physical performance. And so once again, with zebrafish larvae, they injected them with LPS and then the three different substances, and basically watched how far they swam. And you can see at the bottom here, total distance traveled, and on the right-hand side, sort of a tracer graph of how they swam. So the controls swam quite vigorously everywhere. When they were given LPS, however, they hardly moved. They moved very little, as you can sort of see here, and also by this bar chart. And when they were just given donepezil on its own, they swam a bit more, but not really that much. However, when they were given the 2 EVs, they swam considerably more, as you can see by these tracers and also the 2 bar graphs here. And the fat EVs swam the furthest. And that is the end of the paper. Now, hopefully you can see that EVs show great potential for delivering drugs into the brain and seem to have greater efficacy than the drugs given on their own, at least in this case of donepezil. And because they can carry pretty much anything inside of them, they could really help with the diseases of the brain and overall longevity. Now, I really hope that researchers continue with this research, maybe move on to other animals such as mice and eventually to humans. And I really hope that further research like this is and will be taking place in Canada. And that marks the end of this month's review. Please don't forget to like this video, subscribe to our channel, take a look at our online store, and if you wish to help us further our work, donate to the Canadian Longevity Association. Until next month, goodbye.